before we jump into the really interesting and amazing evidence for God related to the origin of life, I want to quickly review how this topic fits into the whole video series that I am working on. So here is a summary of that video series, and these are 7 broad categories of evidence that God has used in my life to strengthen my faith in God and in the truth of the gospel, in the promises of God. So in part 1 of the video series was an overview where I quickly talk about all 7 categories. And then part 2, I talked about supernatural experiences. I'm talking about modern day miracles, several that I experienced, relatively small miracles compared to the big ones in the Bible. So that was part 2. I also talked about some miracles happening around the world. Part 3, multiple reliable eyewitnesses for the resurrection of Christ. What a powerful type of evidence that is for Christianity. Part 4, A and B, this was 2 videos on the moral argument, how morality points to the existence of a great moral lawgiver, and even specifically to Christianity. And in part 5, I'm talking about evidence for creation. Now, it's not that I think that this topic is the most important type of evidence, but it is the topic that I've spent the most time reading and studying about. So I am actually planning on 4 videos on this topic. And the first one, which I've already published, is on evidence and reasoning that is ancient, universal, and reasonable. So throughout history, people have seen evidence for a creator and through the world that he made. So you can look at the world that God made, and this world gives evidence for God. There is a good rational basis for belief in God based on what we see, and you don't have to be a scientist to see it. So this part 5A is not scientific in nature. It's just general humanity and rational thinking. But in the 19th and 20th centuries and continuing today, many scientists came up with theories which they claimed made God unnecessary to explain creation. They're wrong. And to show this, I include videos 5B and 5C. Today's video is part 5C and probably 5D to explain how science provides additional, very strong evidence for God. So I've already published the video on fine-tuning. So you can watch that. That's a type of evidence that I think is very powerful for God. This video is on evidence from the field of biology, and there's so much evidence from biology, but I am focusing specifically on the origin of life. And then, Lord willing, I'll have another video in this section on the origin of the universe. So these 3 are scientific types of evidence for God, and this is the one that we're on today, the origin of life as evidence for God. There is so much evidence in so little time. There is a huge amount of evidence for God in the field of biology. I will try to share some examples of evidence that I find to be exceptionally strong. While the basic arguments here are similar to those many others have made, I think some details I include about how viruses play into this topic are rarely discussed and will be, hopefully, prayerfully, and I bathe every video in prayer. Hopefully, prayerfully, they will be interesting and helpful even to those already interested in this topic who've already read about it. Then at the end of the video, I'll mention some other resources for the study. So I've decided to focus on the issue of the origin of life. There's a lot of other things in biology that we could have talked about here. So basically the issue is this: starting with the type of chemistry and resources one could plausibly find on a planet like the ancient Earth before there was any life. How did the first living cell come into existence? And I'm going to be comparing two competing explanations. So the first explanation is a process guided by a super intelligent and ancient creator whom most people will identify as God. Some people have thought, well, maybe space aliens did it. I don't think that's likely, but the science doesn't tell us. The science, just based on the origin of life, just based on biology, doesn't tell us what type of super intelligent, ancient, very powerful creator. But I think it's probably God. And the other types of evidence verifies that it is God, the Christian God. And I'm sorry if my glasses are a little crooked. Let's see. And then And then the second explanation is an unguided process based on the laws of physics and chemistry in random events, with natural selection working on self-replicating pre-cell chemical systems in a process often called either chemical evolution or abiogenesis. And so these are the two explanations for the origin of life. So here's another way of looking at this. This is what we are trying to explain: how to get from the types of chemicals that could be found on the prebiotic Earth to the first living cell. And I'm not going to get into details on what type of chemicals could be found on the prebiotic Earth. Some people think that maybe there could have been amino acids, for example. Maybe there could have been some sugars. I don't know how likely that is, but it doesn't really affect the issue. You can, even if you assume that those types of things were on the early Earth and available, it's not going to really change the things that we discuss in this video. So this process involves a super massive increase in functional complexity. So a cell is exceedingly complex, and we're going to be talking about that in the video. Even the simplest cell is not actually simple, but it's not just complex. All of that complexity has a purpose. It's like the parts in an airplane. Yes, they're complex, but they all work together to do a purpose. And all the parts in the cell that are very complex are working together to do a purpose. So that's part of what we have to explain. How could that happen? And so as I mentioned, the simplest possible living multiplying cell is not simple at all. So there's two ways I want to share with you of seeing the complexity of simple cells. These aren't the only two ways, but they are the two ways I want to share in this video. One, I want to compare them to nuclear submarines. Now, I had the privilege of serving in the U.S. Navy, and I served on nuclear submarines in the engineering department. So that's probably why I thought of this analogy. And then the second is considering the genome of the simplest known living cell. So that's what we're going to do next. Okay, let's see. So here we have living cells and nuclear submarines, and here are some things that they have in common. Well, let me show you. I thought when I made the picture of the submarine, I thought, oh, people can't really see it. So I'm going to quickly show you this cutaway view of the submarine. The front part is where the control room and all of the weapon systems and the sonar is, and the crew quarters. And then in the back, we have the nuclear reactor and the steam plant, and where we make electricity and where we produce energy to make the submarine go through the water. Okay, so what are some things that living cells and nuclear submarines have in common? Well, nuclear cells have complex hulls, and cells have complex cell membranes, and both have controlled openings that selectively allow the right things in and out. So it's pretty obvious that a submarine needs to have a hull that will keep us safe so that the ocean doesn't come into the submarine and flood the submarine, sink the submarine, drown all the people in the submarine. But there are a lot of protrusions or holes in the hull. When I was on the submarine, before I could be qualified to serve as officer of the deck, I had to learn all of the openings in the submarine, and there's a lot of them. There are openings to get torpedoes in, and then there are openings when we want to fire the torpedoes. There are openings to get food in, and there are ways to get trash and sewage out of the submarine. There are ways to get relatively cool seawater in to condense the steam in the steam plant, and ways to get the hotter water out. Then there are other ways to get water in that we purify. We we evaporate and then condense the water to get the salt out, and then we can drink some of that water, use some of that water in the nuclear power plant. But then some of that water we split the H2O into hydrogen and oxygen to make oxygen for us to breathe if we are under the water for a long time. And so there are many other protrusions through the hull at all. But all of these have to let the right things in, the right things out at the right time, while keeping the ocean out. Well, the cell has a similar situation going on. You need a cell membrane because the chemistry inside the cell is a lot different from the chemistry outside the cell. And if there was no cell membrane, the chemistry that's keeping the cell going inside of it would just diffuse into wherever it was, in a pond or a lake or anywhere, and then the cell would not be able to live. It would not be able to continue to function. But also, the cell needs controlled openings in the cell membrane to let the right things in, nutrients that it needs and raw materials it needs to build all of the chemical machinery that it builds. So it has to let things in, or it wouldn't be able to function very long. And it also has to be able to get out waste products. And so the cell has controlled openings that are like the types of molecular machines that selectively allow the correct things in and get the right things out. So that's one way that cells and submarines are similar. Both have complex energy production systems. Obviously, a nuclear reactor and the associated steam plant is very complex. But wow, the cell has a very complex energy producing and management system. One piece of that system is called the ATP synthase. You can Google that. You can find videos on YouTube of how that thing works. It's a molecular machine. It's amazing. But that is just one in a whole series of complex molecular structures that allow the cell to produce energy in the form that it needs. And so that's similar to what's going on in the submarine. Both are stuffed full of machinery. Boy, you walk through a nuclear submarine and everywhere there's machines. There's machines to control the atmosphere. There's machines to pump the water that's needed for to cool the nuclear reactor and to cool different items in the submarine. There's air conditioning machines because the steam plant makes a lot of heat, and you really it's not just a luxury. You really need the air conditioning, or it'll become extremely hot inside the submarine. And there's all the machines that make electricity, and there's the machines that make the submarine go forward. And there's just a lot of machines inside a submarine. There are also a lot of machines inside a cell. Even the simplest cells have a lot of molecular machines, and they really are. It's accurate to call them molecular machines. Again, I encourage you to Google or look on YouTube for molecular machines, and you'll find great videos and pictures of these molecular machines. And there's lots of them in every cell, even in the simplest cells. And then both submarines and living cells have digital information storage and processing systems. So, we use computers for the fire control system, to control torpedoes or missiles that we fire, for sonar processing, for a lot of things. And the cell has a way to store digital information in the DNA, and then it has other types of molecular systems. These could also be accurately called molecular machines that read and process and use that information to build new proteins and to run the activities of the cell, and when it multiplies, to make a new cell. So they both have digital information and processing systems. Now, it's true that nuclear submarines can do some things that cells cannot do, but it is also true that cells can do something that neither submarines nor any other man-made machine can do. Cells can self-replicate, and we are going to be talking about more about self-replicating as we go through this topic. But just think about it. Imagine how amazing it would be if you had a submarine that could grow and get bigger and then split in two and make two baby submarines that grew into adult submarines. Obviously, no submarine can do that. No man-made machine, robot, factory can do that. Maybe one day people will make a machine that can do that. So far, no machine can. Or system of machines can do that. But every living cell, every blade of grass, every tree, every board, and every fish in the ocean, all living things are the types of things that can self-replicate. So now I want to talk about the other way to look at the complexity of a cell. And here I want to focus on the very simplest cells. So the submarine analogy is helpful to begin to get a feeling for the complexity of a simple bacterial cell, but a more exact measure was produced by a team of researchers led by Craig Venter. In 2016, they published the results of their research in an article entitled Design and Synthesis of a Minimal Bacterial Genome in Science magazine. And you can look at that article. It's a great, interesting article to read. All cells in all living creatures contain DNA. DNA is a very long molecule that has a string of bases symbolized by the letters A, C, T, and G. The order of the letters, or in other words, the order of the different bases, form a code that contains directions for assembling proteins and other types of instructions for the cell. And the proteins make the parts that make these molecular machines I was talking about. And there's thousands of different kinds of proteins. So if cells in their parts, like proteins and RNA strands, only needed very short, specific strings of DNA code, and if somehow DNA code was being produced randomly on the early Earth, perhaps you would get the code you need. But cells do not need very short strings of DNA code. Craig Venter and his team, using a combination of theoretical and experimental methods, worked hard to produce a living cell with approximately the smallest possible genome, the least number of DNA base pairs in the DNA of the cell, in order for the cell to still be able to live and grow and multiply. Now, it's amazing how they did this. It was a lot of work that they put into this. Again, if you read the article, it's just fascinating. And theoretically, so they began with a cell that had one of the smallest genomes from nature. And these types of cells live in the guts of mammals. And that helps them to be small because they can use a lot of the chemicals from their host to keep themselves alive. So they don't have to make all of their own chemical parts because they can't live outside like just in a pond. They have to live in the gut of a mammal. But then they took this very simple cell and they said, can we make it simpler? And I'm going to give a very simplified version of what they did. Theoretically, they said, people have already done lots and lots of research on cells, and we know a lot of the things that they need. They need a way to make DNA, copies of DNA, to use the information from the DNA and transcribe it over to RNA, and then there has to be a way to use the information in the RNA to build proteins, and we need proteins to manage the energy system. We need to be able to make the cell membrane and have including the needed openings in the cell membrane. So they had a list of things they needed. And then there were things they thought that even though it was beneficial for the cell to have them, the cell could do without. And so they cut out the parts of the DNA that coded for the parts of the cell that were optional, that they thought were optional. And some of those parts they found out, oh, hey, it's not really optional. And they found it out because when they cut that out of the DNA, the cell couldn't live. And so they did that until they got approximately the smallest possible genome. And based on both theoretical and experimental results, they have a good argument for this being approximately. They're not claiming they got exactly the smallest possible genome, but they think they got in the ballpark. So that genome contained 531,000 base pairs worth of code in 473 genes. Genes are sections of the DNA that code. Usually they code for proteins, but they can also code for functional RNA strands. And so that's 531,000 of those ACTG letters was the minimum, approximately, that they think any living cell can get by with. But the first living cell probably needed a significantly larger genome. Remember, these things were living, the cell that they started with lives in the guts of mammals, but then when they were doing these experiments, they had them in a rich growth medium so that the cell had a lot of help surviving that it would not have if it was by itself in an ocean or in a pond somewhere. Here's two statements from the article. Because our minimal cell is largely lacking in biosynthesis of amino acids, lipids, nucleotides, and vitamins, it depends on the rich medium to supply almost all of these required small molecules. So on the ancient Earth, the first cell probably would have had to make a lot of its own molecules. In addition, because of the rich growth medium that supplies almost all of the necessary small molecules, many genes involved in transport, catabolism, proteolysis, and other metabolic processes have become dispensable. So in other words, they're saying we were able to get rid of things that in the wild, the very first cell probably would have needed. Now, they were not trying to make a cell that was as similar as possible to the first cell. They were just trying to make a cell that could live and multiply, get in a friendly environment, a growth medium that had the smallest genome possible. And there was a lot of experimental knowledge and scientific knowledge to be gained from this. This really wasn't an origin of life experiment, but it has a lot of origin of life implications. So the genome contained 531,000 base pairs worth of code in 473 genes. It is utterly, completely impossible to generate that much functional information by a random process. Origin of life scientists know this, and as far as I know, none of them think this is what happened. Now, before we discuss what they think did happen, let's look at an analogy to help us understand why generating that much information is impossible. And I'm going to use a chocolate chip cookie analogy. I was recently reading a new book put out about science and faith, and Doug Axe has a great article in there where he uses a cookie analogy. Some of the points he makes with it are similar to this, and some are a little bit different, but it kind of inspired me, I think, to use this chocolate chip cookie analogy. So here's a recipe for chocolate chip cookies that I found on the internet, and I want us to focus on just one line of instructions from this recipe. So here we go. Beat butter, granulated sugar, brown sugar, and vanilla in a large bowl with mixer until creamy. So this contains 76 characters, not including punctuation or spaces. So just to simplify, we're not going to include punctuation and spaces. And the vast, overwhelming majority of character strings of roughly equal length are useless nonsense. So with my eyes closed, I just randomly typed some letters. If you did this, you'll find that you always get nonsense. You're never going to get a sentence that makes sense. And certainly you aren't going to get a line of instructions that would be useful for making chocolate chip cookies. In fact, the chance of typing those letters randomly, if you had a keyboard of only the 26 letters and no capitals, would be 1 in 26 raised to the 76th power, which is 3.4 times 10 to the 107th, which is a mind-blowingly huge number. If there were a trillion Earth-like planets in a galaxy, that's not realistic, but if there were, and they were all covered with monkeys, 1 monkey per meter squared, and I'm including the ocean, even though monkeys can't sit on the ocean, and each monkey had been typing 176 character string every minute since the universe began. So I picked monkeys because they don't know language. They're just randomly typing. How you would convince them to do this, I'm not sure, but if they did, they would still be very unlikely to type that sentence even one time, even if they'd been typing since the universe began, and all of them were typing on a trillion planets covered with monkeys. You can check my math if you want. In fact, the monkeys don't even come remotely close to having a chance of typing this sentence. There are only approximately 1 times 10 to the 80th atoms in the observable universe, but you only have a 1 in 3 times 10 to the 107th chance that any given random sentence will match this one that's in the recipe. Now, so it's impossible to get one medium-length line of instructions correct. How much more to get over 500,000 DNA characters correct? And it does work like an alphabet, but it's an alphabet with only 4 characters instead of 26. To get the equivalent amount of information with a 26-letter alphabet, you would have to divide the number of DNA characters by approximately 3, and I'm not going to explain the details of that. But the point is, even if you said 100 and whatever it would be, about 160,000 or something, there's no, I mean, you can't even get 76 characters. It's just utterly, utterly impossible to get that. Now, here's a possible objection. But aren't there many different DNA combinations of 500,000 base pairs length that would work in a living cell? And the answer is yes. There's probably trillions or quadrillions. I don't know how many, but a whole lot. But the combinations that would work are way too, or that should be TOO, sorry about that, way too extremely, super incredibly rare to be found by chance. There's been good research to show that that is the case. So, of course, origin of life scientists don't claim that a fully functional cell popped into existence by chance. No one, as far as I know, thinks it happened that way. Instead, they speculate that something like this happened. A process of chemical evolution where thousands of relatively small steps happened in between the initial chemicals and the first living cell. So they said, okay, you can't get there in one big step. The probability of getting the DNA code right, and that's not the only thing you need to get right, by the way, but the probability of getting that is so fantastically, super incredibly small, there's no way you could do that. But what if there were lots and lots of teeny little steps, and each step could be done? What if? So this is what they have in mind. For this to work, at the very least, each step must consist of a self-replicating molecule or system of molecules. If it's not self-replicating, it might be interesting, it might be fun to study, but it would be useless in progressing in chemical evolution towards the first living cell. They have to be increasingly complex on average so that you eventually get to the complexity of the cell, and they have to be increasingly cell-like because otherwise you would never get to a cell. So that's what's needed, and you need a lot of steps. because if any single step was too big, it would be impossible for it to happen by natural forces and random changes to the system that could be preserved by natural selection. So many thousands of steps would be needed, and all of them have to fit these criteria. Now, here's the problem. It's not the only problem, but it's one really big problem. These steps have to have self-replicating molecules or systems of molecules. What do we mean by self-replicating? This is important. By self-replicating, I mean a molecule or system of molecules that completely replicates itself using material that would have been available. And so that definition is going to be important, and it just makes sense that that's what had to happen if the naturalistic explanation for the origin of life is correct. That something like that had to happen. Now, many thousands of such increasingly complex self-replicators would have been needed. How many have been found so far in nature and or created in a lab? How many systems of self-replicating molecules short of a complete living cell have been found in nature or made in a lab? And the answer is 0. None at all. If such self-replicators ever existed, it is odd that we have not found them. We would expect that these systems might consist of things like RNA, DNA, proteins, or systems with some type of membranes, especially the systems that were getting close to the cell, like maybe the last 1,000 systems, because eventually they have to have a cell that has all of these things. And here's what's interesting. Huge numbers of systems that are simpler than cells and that have DNA or RNA and proteins and membranes exist in nature. They are called viruses. And people have been thinking a lot about viruses. All viruses have either DNA or RNA in them. They have to in order to make new copies of the virus and multiply. Many of them also have come prepackaged with some proteins, some molecular machines that are going to help them when they invade their host, and most of them, or at least very, very many of them, have a simple type of membrane. It's not a complex membrane like a cell has that can allow certain things in and out, but it's a protective membrane, and the membrane sometimes also helps the virus to fool the cell and to sneak into the cell. Now, here's the interesting thing about all of these viruses. Well, one interesting thing is that there's a huge number of them. They're all estimated to be 10 to the 31st. That is huge. That's more than a trillion. That is huge. They're estimated to be 10 to the 31st virus particles on Earth, with probably well over 1 million different types of viruses. As far as we know, none of them can self-replicate. None of them. We have not found a single example of a virus that can self-replicate. They all hijack the replication machinery of living cells in order to replicate. So viruses, certainly all of the viruses we know of would have been useless as a step in the process of getting from simple chemicals to the first cell, because the viruses need a cell in order to replicate. And by the way, viruses don't just infect people. They infect all kinds of animals. They infect plants. There are even types of viruses that infect bacteria. So there was just a huge number of viruses. Now, the simplest explanation for the fact that viruses cannot self-replicate is that no organic system short of a living cell can self-replicate. Because if anything could, you would think it would be a virus, because it has a lot of the pieces that would be helpful, and yet none of them can. And you would think, because cells have defenses against viruses to keep them from getting in and hijacking their machinery, boy, you would just think it would be super useful if a virus could get the ability to self-replicate and didn't always have to get inside a cell. But none of them, as far as we know, can. Maybe one day somebody will find one that can. I don't expect that to happen. But based on what we do know, none of them can, what we know so far. And I don't think that's going to change. It certainly seems unlikely that thousands of such systems existed in the past, but now 0 such systems exist. What about self-replicating molecular systems produced by scientists in a lab? So if you Google self-replicating molecules or self-replicating RNA or self-replicating molecular systems, you will see some articles that the article title and sometimes the way it's written makes it sound like scientists have created self-replicating molecules in the lab. They have not with the meaning of self-replication that I gave, which is the type of self-replication that would be needed for the origin of life. I've looked closely at the best claims of self-replicating systems of RNA and found that none of them are truly self-replicating because either, one, they do not fully self-replicate. So the copies they make are only part of themselves, and those copies cannot make other copies. They don't copy enough of themselves to keep the thing going, or, this one is more complicated, they need to continuously be given already mostly assembled parts to work, so that in reality 99% or more of the replication is done by the scientists. One of the articles that was suggested to me as one of the very best examples of molecular self-replicating system, I really read it carefully and studied it, and it turns out that what the scientists did, it was a good experiment. It just doesn't produce a system that would be one of the steps needed in the origin of life. Because what they did is they had a RNA molecule that could cause, and it had to have a very specific sequence. There's more than one sequence that would work, but the sequences that would do this are super extremely rare. So this sequence was designed by the scientists, a sequence of base pairs, because RNA has base pairs similar to DNA. And what it could do is it could cause two halves, two shorter sequences of RNA to join together. But the only way this would self-replicate is if those two shorter sequences each had half of the exact sequence of the mother molecule. And that is so extraordinarily, super incredibly unlikely to happen that even if you had an ocean full of RNA molecules, it's exceedingly super unlikely that those three pieces, the mother and the two halves, would ever find themselves and get together. And if they did, it would make one copy and it would stop, because you have to keep feeding it the halves in order to work. So this is not a origin of life relevant example of self-replication. And I don't know if they'll ever succeed in making a really truly self-replicating system of molecules outside of a living cell. So far they have not, and the odds are not in their favor. I don't think it's likely that we will make that. By the way, if really intelligent scientists, after years and years of work, many different scientists attempting this, if somebody finally makes it, what it shows is that intelligent designers can make such a system. It would not prove that nature, without any scientists on the ancient Earth, could do the same thing necessarily. So far we have looked at several major problems for a completely naturalistic origin of life. One, the functional complexity of even the simplest cell. Oh, let me turn myself back on here. The functional complexity of even the simplest cell and the vast amount of functional information that is needed. So you can't get there in one step. But what if you try to get there in lots of little steps? Well, the second big problem is the complete lack of any known self-replicating systems other than cells, despite a vast number of viruses that are the types of things that if anything could self-replicate outside of a cell, you would expect it to be something very, very, very similar to a virus. But none of them can do it. And efforts to produce self-replicating systems in labs so far have not gotten there. So these are two huge problems for a naturalistic explanation of life. There are other big problems that I will just mention quickly. The chicken and the egg problem on a massive scale. So what if you got the correct sequence of nucleotides in either an RNA molecule or DNA molecule, and it was floating somewhere in a nice little pond or near a vent down at the bottom of the ocean or somewhere? Would that molecule then go on to produce a cell, since it has the instructions for a cell? And the answer is absolutely not. It is useless without the molecular machinery to read those instructions and to use those instructions to build the cell and to provide the type of controlled energy system to operate the machines to use those instructions to build the cell. Where do all those molecular machines come from? They come from the instructions in the DNA. How is the DNA made? It's made by those molecular machines. So you can see this is a chicken and the egg problem on a supermassive scale. So that's a big problem. The instability of RNA is a big problem. I mentioned RNA for two reasons. Eventually, these pre-cell systems that are hypothesized would have to include RNA because the simplest living cell certainly had RNA in it. But also, the most popular scenario for chemical evolution is still the RNA world, is what people call it, saying that before there were cells, there was RNA that either the molecules by themselves or in systems of molecules could self-replicate. But RNA is not a stable molecule. It doesn't last for a long time. DNA is way, way more stable, and that's why it's used for long-term storage in cells. But RNA is used for the temporary job of building proteins. And so that's and RNA is unstable in water. And it doesn't last a long time. It lasts long enough in a cell to do its job. But if you got some RNA that was useful, which I think is impossible, but if you did on the early Earth, it wouldn't go on to make more and more and more steps. It most likely, almost certainly, overwhelmingly, the most likely thing is it would just disintegrate wherever it was at. That wouldn't lead to the origin of life. The RNA stickiness problem, you can Google this. RNA sticks to a number of other, it sticks to other RNA, and this can help it to be used as a template in replication, even though we haven't got it to do full self-replication. But then the things tend to stay stuck together, and that would end the process if they stayed stuck together, and they do tend to stick together. You can Google this, and you can find this problem. Chemical synthesis difficulties for the chemistry needed for life. So a lot of what I've been saying kind of assumed that there could have been RNA floating around, and you just had to get the right sequence, which is like super impossible. But it turns out that there probably was not any RNA floating around, or any of the right types of sugar, or any of the proteins that would have needed. These types of things are incredibly difficult to synthesize outside of cells in a prebiotic setting. James Torr, who is a world-renowned synthetic chemist, has a series of excellent videos on this topic. You can easily find them if you look for them on the internet. And then the transition from RNA as an enzyme to the RNA DNA carrying encoded information for proteins. So in cells, there are some RNA strands that work as enzymes. They catalyze some chemical reactions, or they're involved in that together with proteins sometimes. But the main job of DNA and RNA is coding information, not working as enzymes, because the RNA is not as well suited to be an enzyme as proteins, not even remotely as well suited. But for the origin of life scenario, chemical evolution to work, a lot of the scenarios assume that you would have RNA working as enzymes, and then later that would transition to what the cell does, which is almost all the enzymatic activity is carried out by proteins. But here's the problem. Let's say an RNA is doing a certain function, and in order to do that function, it has to have a certain sequence of base pairs. That's what determines that the RNA can do that function. That sequence has no relation whatsoever with the coded sequence of base pairs for a protein that would do the same function. Now, that's kind of a complex thing to think about. You might have to go back and listen to it 2 or 3 times, but it's a huge problem for chemical evolution, origin of life scenarios, and more. There's a lot more issues here. So intelligent beings can design and produce complex systems with many parts requiring lots of information. Intelligent beings make submarines and airplanes and spaceships and computers and iPhones, but we've never seen natural forces and random events making complex systems with many parts requiring lots of information that do some kind of a function, and that's what we see in life. So I think intelligent design is a much better explanation. Now, is this a God of the gaps argument? Some people will say, oh, this is a God of the gaps argument, and they say, look, there's even a gap. You even have a gap here in a lot of your pictures, a gap between the simple organic molecules and life. Well, this is not a knowledge gap in my slides. This is showing a complexity gap, a functional complexity gap. So those are 2 totally different types of gaps. The argument that God is a better explanation than purely natural processes accounting for the origin of life is not based on what we don't know, it's based on what we do know. So I want to give an analogy here. Let's imagine that there were a couple of people, 2 guys, let's say, and for some reason, just go along with this for the sake of the analogy, they knew nothing about the whole Apollo space program and people landing on the moon. That's not realistic today, but let's say that for some reason, maybe they both had really super weird brain injuries that caused them to forget all about that or something. But they remembered pretty much everything else in life. And they found out that there was an American flag on the moon. Maybe they saw a picture of just the American flag on the moon. And they both agreed that a person must have put it there, but then they got into an argument about how the person got up to the moon. And one person said, well, I think it was done with stairs. We don't know of any set of stairs that goes that high, but obviously a person can't jump all the way from Earth to the moon. But maybe if there were lots of little steps and the steps reached up to the moon, that they could get to the moon that way. And just because we've never seen any steps that go that high doesn't mean that steps never could go that high. And the other person said, no, I think rocket ships did it. Now, this person doesn't know that rocket ships went to the moon, but he knows that rocket ships go up to the International Space Station. He knows that rocket ships take probes as far as other planets. So he says, I know that rocket ships are capable of doing the types of thing that needs to be done here, which would be, getting something, in this case, a spaceship with people in it, up to the moon. So I think the rocket ship is the best explanation. Now, even though there's a gap here, and there's even a gap in knowledge, because you can say, well, we don't know, we don't actually have any direct evidence. These 2 people don't have any direct evidence of how this was done, but we know about steps. And there's lots of things we know about steps that tell us that it would be practically impossible, just for many reasons, it would be practically impossible for somebody to walk up steps and get to the moon. But we also know about rocket ships. We know that it would be practically possible for a rocket ship. That's the type of thing a rocket ship is good at, would be getting to the moon. And this is the same type of argument that I'm making with the origin of life. There are many reasons that it is practically impossible for a natural, unguided process to produce the first living cell. But producing a living cell is exactly the type of thing that intelligent agents can do. Now, scientists have not been able to make a cell from scratch yet, but if there was some type of an intelligent agent who's much smarter than scientists and more powerful and was around a long time ago, was ancient, it's very reasonable to think that he could make the first living cell. And of course, I believe that is God. So this is not an argument based on what we don't know. It's an argument based on what we do know about natural processes, and about intelligent designers, and about the nature of living cells. And so all of this information we do know says that it's vastly more likely, super vastly more likely, that an intelligent agent is responsible for the origin of life, not an undirected natural process. The emergence of life on Earth appears to be a miracle, something that God did, is what I mean by that, and that nature wouldn't be able to do on its own without intervention from God. Now, the word miracle can have different meanings, but in this case, that's what I mean. And I'm not the first person to think so. Francis Crick, who was one of the men who, he got a Nobel Prize related to discoveries with the DNA molecule. He discovered its structure, and they also discovered basically how it works, the sequence hypothesis. And he said, an honest man, armed with all the knowledge available to us now, could only state that in some sense the origin of life appears at the moment to be almost a miracle. Now he says almost a miracle. Well, if you don't believe in God, then that's probably the most you can say is almost a miracle. And then Eugene Koonin, who is a prestigious biologist, he said, a succession of exceedingly unlikely steps is essential for the origin of life, from the synthesis and accumulation of nucleotides to the origin of translation through the multiplication of probabilities. These make the final outcome seem almost like a miracle. And that's what I think it is. I think that God did it. That is by far the best explanation. I want to give you some resources for further study. I love reading about this. I've read all of these books. I recommend all of them. There are other books, and even other books that I've read that are related to this topic, but I'll just give you these six. Signature in the Cell by Stephen Meyer, probably the book I would recommend most on the issue of origin of life. Some of these books are about biological evidence for God, but not the origin of life. They have to do with, okay, once you have the living cells, how do you get to flowers and butterflies and dolphins and people? So Darwin's Doubt would be in that category, focusing on the Cambrian explosion. Excellent book also by Stephen Meyer. The Stairway of Life is another origin of life book that looks at, I believe they have 12 steps, and these steps actually kind of all had to happen at once. They explain this, but we can simplify the process and think of them as 12 steps that would be necessary to get from chemistry that could have been on Earth without living things up to the first living thing, and why intelligent design is a far better explanation for that than a natural process. And then 3 books related to why intelligent design is a better explanation for the variety of life than evolution. 3 books by Michael Behe. I have a blog post where I give a summary of kind of the main points of each of these 3 books. They're excellent books. Darwin's Black Box, The Edge of Evolution, and Darwin Devolves, all by Michael Behe. And these books are all well written and well argued, so I highly recommend them. Now, I want to close with a devotional thought. This is less apologetic in nature and more devotional, even though it's related to the same topic we have been discussing. So here we go. In this video, I've focused on the simplest life, which is not simple at all. But how about you? Your body consists of approximately 37 trillion cells, and every one of those cells contains some 3 billion base pairs of DNA, and all of it works together to produce a body that, when healthy, can walk and dance and see and talk and think and study and write and read and learn about the evidence for God and read about His love and grace and salvation and kneel down and worship. I pray that God will bless this video and will bless everyone who watches it.